Bismillahirrahmanirrahim. Assalamu alaikum, second year. How are you? Hope you are good. Now we will continue with the development and aging, chapter number 21, and today is the lecture number 2. Growth and related features describe the role of nucleus and cytoplasm in controlling development through experimental narration. Describe the role of nucleus and cytoplasm controlling development. Brief overview of the work done by Spemann in the discovery of induction. Define organizers and differentiate between primary and secondary inductions. And human embryonic development. A brief overview of this article. Now, growth. What is growth? It is the most obvious phase of development, which is characterized by increase in size and mass of an organism. It begins soon after fertilization and remain continue throughout the development. Organogenesis is almost accomplished in 8 weeks. By the start of 3rd month, human embryo is called fetus. The rest of the development comprises only growth and maturation of organs and systems. Control of development. How does the developing embryo generate the multitude of many cell types of a complete multicellular organism from the starting point of a single diploid nucleus of a zygote? Many work done by embryologists in 19th century, and we have very prominent scientists or embryologists in this regard. Here we have the two names, the Hans Driesch experiment and Hans Speamann's experiment. These two are very prominent embryologists in the field of control of development, and here we will discuss the experiments of these two one by one. Hans Driesch's experiment in 1892 discovered that if he mechanically shook apart a two-celled sea urchin into separate cells, both half embryos developed into normal larvae. Driesch concluded that both cells contained all the genetic information of the original zygote. Still, this experiment did not settle the argument because many embryologists believed that even if all cells contained complete genomes, the nuclei might become progressively modified in some way to dispense with the information they do not use in forming differentiated cells. Here is the graphical representation or the photographic representation of Hans Driesch experiment. Here is the Pluteus larva, and by the separation of these this 4-celled stage, you can produce larvae of one from each cell. Here we have the Hans Speamann's experiment, figure of Hans Speamann's experiment. To describe this experiment, we have a new approach with Hans Spemann, and he tests the Rox-Weismann's hypothesis. Spemann placed minute ligatures of human hair around the salamander's zygote just as they were about to divide, constricting them until they were almost but not quite separated into two halves. The nucleus lay in one half of the partially divided zygote. The other side was anucleate, containing only cytoplasm. The zygote then completed its first cleavage division on the side containing the nucleus. the nucleate side remained undivided. Eventually, when the nucleated side had divided into about 16 cell stage, one of the cleavage nuclei would wander across the narrow cytoplasmic bridge to the anucleate site. Immediately, this side began to divide with both halves of the embryo containing nuclei. Spemann drew the ligature tight, separating the two halves of the embryo. He then watched their development. Usually, two complete embryos will be resulted. This showed that the single nucleus selected from the 16 cell stage embryo contained a complete set of genome. All were equivalent. Sometimes, however, Spemann observed that the nucleated half of the embryo developed only into an abnormal ball of belly tissue, although the half that received the delayed nucleus developed normally. Here, question arises, why nuclei of 15 cell out of 16 failed to develop and the one which was transferred to non-nucleated site resulted in the embryo development? For this question, we have an answer. Spemann discovered, depending on the position of the gray crescent, what is gray crescent? The pigment-free area that appears at the moment of fertilization. If one half of the constricted embryos lacked a part of gray crescent, it would not develop. Instead, will change into undifferentiated mass of cells. Here is the animal hemisphere, vegetal hemisphere, animal pole, then the point of sperm entry, the vegetal pole. Here we have the gray crescent. If it is absent, then the development is disturbed. Conclusion. Spemann's delayed nucleation experiments served as compelling evidence for two important conclusions. One is that all cells contain the same nuclear information, thus disproving the Rox-Weismann hypothesis. And second, cytoplasm in the area of gray crescent must contain information essential for normal development. If all nuclei are equivalent, then what causes some cells to develop into neurons while others develop into skeletal muscles? In most animals, excluding insects, listen, there are two major ways by which cells become committed to particular developmental fates. First, the cytoplasmic segregation of determinative molecules during cleavage, and second, interaction with neighboring cells, inductive interactions. All animals use both mechanisms to specify different cell types. However, in some animals, cytoplasmic specification is dominant, whereas others rely predominantly on inductive interactions. From all these experiments, it was concluded that both nucleus and cytoplasm play an important role in the development. Nucleus contains all the genes which determine the characteristics of the individual, while cytoplasm is involved in the selection of genes in the particular cell types, as it contains some components called the morphogenetic determinants that are involved in the expression of genes. Now, the embryonic induction. The capacity of some cells to evoke a specific developmental response in others is called embryonic induction. It is a widespread phenomenon in development which was reported by Hans Spemann and held Mangold in 1924 in a series of classic experiments on amphibians embryo. Here we have the Spemann's experiment. In this experiment, when a piece of a dorsal blastopore lip from a salamander gastrula was transplanted into a ventral or lateral position of another salamander's gastrula, it is invaginated and developed a notochord and somites. It also induced the host ectoderm to form a neural tube. Eventually, a whole system of organs developed where the graft was placed and then grew into a nearly complete secondary embryo. This creature was composed partly of grafted tissue and partly of induced host tissue. It was soon found that only grafts from the dorsal lip of the blastopore were capable of inducing the formation of complete or nearly complete secondary embryo. This area corresponds to the presumptive area of notochord sometimes, and somites are there, and the precaudal plate. It was also found that only ectoderm of the host would develop a nervous system in the graft, and that the reactive ability was greatest at the early gastrula stage and declined as the recipient embryo got older. Spemann designated the dorsal lip area the primary organizer because it was the only tissue capable of inducing the development of secondary embryo in the host. He also termed this inductive event primary induction because he believed it to be the first inductive event in development. Subsequent studies showed that many other cell types originate by inductive interactions, a process called the secondary induction. For example, cells of the neural plate induce neural crest in the embryo. Here you can see the Speamann's experiment in the form of figure. We have the donor portion or the donor embryo, dorsal lip of the blastopore. Then it is induced in, it is grafted in the developing embryo, and this is primary induction. Okay, and then we have the secondary invaginations, and we have two types of nervous setups. Two neural tubes. One is the original one and the other one is the induced in the already growing embryo, and here we have the two embryos due to the induction of primary organizer. Here we have a video to understand this experiment. We will play it.
Hans Spemann and Hilde Mangold were pioneers of developmental biology. They showed how the pattern of the embryo is created by interactions between one group of cells and another. In 1924, they made a famous discovery. They found that a small piece of tissue called the organizer, taken from a specific site in the early frog embryo and transplanted to another embryo, could control the behavior of neighboring cells and direct the formation of an entire body axis. The key experiment is reenacted here by a modern developmental biologist using the frog Xenopus laevis. Two Xenopus embryos are maneuvered under the dissecting microscope. The embryos are beginning to gastrulate. The blastopore, where cells are tucking into the interior, is visible as a dark crescent in the embryo on the left. The dorsal lip of the blastopore contains the organizer cells. With a pair of forceps and a fine tungsten needle, a block of organizer tissue is cut from the embryo on the left. Using a hair plucked from a human eyebrow, the block of tissue is gently pushed into a site on the ventral side of the other embryo. An hour later, the graft has healed into the host embryo, and the organizer cells have been integrated at an ectopic site. 2 days later, the host embryo has developed into conjoined twins. The grafted organizer has caused the host cells around the graft to form a second body axis, complete with central nervous system, eyes, somites, and other structures.
Now the human embryonic development. Embryonic development begins with fertilization. After fertilization, zygote undergoes changes which lead the formation of fetus and then ultimately an adult. Fertilization of an egg by a sperm normally occurs in proximal part of the oviduct. As the zygote passes down oviduct, it undergoes cleavage which leads to the formation of blastocyst. Pregnancy is usually established when blastocytes is implanted into the endometrium, and you have seen this in the video in the previous lecture. If the implantation is successful, then the embryo begin to secrete human chorionic gonadotropin, which is known as HCG, and this hormone forces the corpus luteum in the ovary to continue to secrete progesterone and thereby maintaining the endometrium and inhibiting FSH production. The corpus luteum and FSH you have done in previous chapter. The chorion, one of the membrane that later grows and surrounds the embryo, develop villi or the projections that burrows into the endometrium. It is also thought that fertilization and conception are synonyms with establishment of pregnancy. However, at this stage, the zygote is free floating in the womb. For pregnancy to be successfully achieved, the embryo must implant in the endometrium of uterus. Sometimes conception is not followed by implantation, hence no pregnancy is established. The human gestation period, the duration of pregnancy, average 280 days from the time of mother's last menstrual period to the birth of the baby, or 266 days, about 9 months from conception. Human pregnancy can be divided roughly into 3 trimesters, each approximately 3 months long. The first trimester is from the last menstrual period, LMP, to 13th week, and the second trimester is from the 14th to 27th week, and the third trimester is from the 28th week to 36th week. Okay ji, this is the end of this lecture. Inshallah, we will continue this chapter in lecture number 3. Till then, Allah Hafiz.